So welcome back, everyone. Next session is going to be focused on beyond the kidney. So I'm going to turn it over to the moderators to lead the discussion and introduce the speakers.
Thank you so much for the invitation. We have exciting talk today covering topics such as polycystic liver disease, the role of cardiac polycystin, polycystin regulation of ciliary extracellular vesicles, and nutrient sensing mTOR pathway in renal cystogenesis. My name is Francisco Altamirano, and I'm an assistant professor at Houston Methodist, and I'm happy to introduce my co-chair for this session, Dr. Cheng Song, who is an assistant professor at the University of Alabama at Birmingham. So Dr. Song, would you like to introduce our first speaker?
All right, thank you, Francisco. So it is my great pleasure to introduce the first speaker for this session, Dr. Scott Waddell. So Dr. Waddell is the postdoc researcher at the MRC Human Genetic Units in the University of Edinburgh. And his current work focuses on the interaction between the immune cells and the cystic cells in the liver. And today he will let us know about his work aimed at uncovering the targetable mechanism for liver cystic growth. Welcome, Scott.
Thank you very much. I assume you can hear me okay?
Yes.
Perfect. And you can see my screen?
Yes.
Yeah.
All right, thank you. So thank you for that kind introduction, and thank you for giving me the opportunity to present my work today. And like we said, we're going to switch focus onto the liver now. So we know that the liver is comprised of two major epithelial cell types. We have the hepatocytes, which make up the bulk of the liver, which makes up the parenchyma, which is highlighted in gray by the schematic. And we also have the biliary epithelial cells, or cholangiocytes, which make this bile duct network shown in blue. And patients with polycystic liver disease have 10 or more fluid-filled cysts throughout the liver, which are made up of the same cell types as this biliary network. So we know polycystic liver disease is the most common extrarenal manifestation in autosomal dominant polycystic kidney disease. And unlike the kidneys, the liver function is typically preserved with patients with polycystic livers, but they suffer from a poor quality of life due to several associated complications due to the fact the liver is increasing in size with age. And the only curable treatment for polycystic liver is transplantation. However, this is reserved for patients with the most extreme cases. And I'm showing an example of that in the bottom right, where we have a CT image where we can see that the liver is spilling throughout the abdomen, and we see cysts all throughout the different lobes of the liver. Pharmacologically, there are no approved therapeutics globally for polycystic liver disease, but we do have some targets, and a lot of these are in common with what we know from renal cysts. So we have elevated cyclic AMP levels, and we have mTOR, among other things that we are familiar with. But for polycystic kidneys, where we know that tolvaptan is FDA approved in certain cases, we know that that's going to bring down the fluid secretion of the renal cysts, and it also acts by preventing or for decreasing cyclic AMP levels. In the liver, we have a parallel mechanism where we can give somatostatin analogs. These have been approved up to phase 2 in clinical trials. And again, they bring down the cyclic AMP levels and the cystic biliary epithelial cells. However, only some patients benefit from these. And even the patients that do benefit after treatment withdrawal, we sometimes see the acceleration of cyst growth again after treatment withdrawal. In addition to this, patients develop hyperglycemia and diabetes with this treatment as well. So we need to do a bit more to understand what are the best targets in polycystic liver disease and how can we control cyst growth in the liver. So I don't need to explain to this audience the implicit role of primary cilia in cystogenesis. On the left, I'm just showing you a mouse bile duct in red, standing for keratin 19. And in the lumen, we can see that there are these primary cilia that project in cyan. And this is similar to what we know from the kidney tubules. And we're all familiar with the fact that the most common genes that cause autosomal dominant polycystic kidney disease, the polycystins, we know that these localize to the primary cilia, and we know that over 90% or 95% of patients have mutations in these genes, and over 90% of patients will have both polycystic kidneys and liver. By comparison, we have isolated polycystic liver disease, which is a much rarer disease that affects 1 in 100,000 globally. And we know about 50% of the causative genes. Most of these encode proteins that localize to the endoplasmic reticulum. And work from the last 10 years from the Somno lab has shown that when we lose function in these proteins, it affects the processing and trafficking of the polycystins into the primary cilium. And finally, I just want to highlight ciliopathies, which are these multisystemic diseases, where we don't always see clinical presentation of liver abnormalities, whether that be due to the fact that they're not always there or because it is masked by abnormalities in the kidneys, the lungs, and things like intellectual disability that make the liver overlooked. But in some patients, we do see reports of cystic dilation of the bile ducts and ductal plate malformations, so the inability to form a proper biliary network during development. So we took a bit of a broad stroke approach, and knowing that you have ciliary dependent and independent mechanisms of cystogenesis, we thought we'd just look at the role of cilia. And work published previously from the Pleasantine Mills group here in Edinburgh has shown that when we delete the essential cilia gene WDR35 or IFT121, we are unable to form functional holo complexes of the IFTA complex. And in cells that have this mutation, they form these rudimentary organelles on top of the cell. So we're able to form IFTB complexes. They localize into what would be the ciliary compartment. But due to the lack of IFTA complexes, we are unable to elongate beyond a rudimentary organelle on top of the apical membrane. In addition to this, we are unable to localize membranous cargo into this compartment, so we don't see any smoothing receptor localized to the mutant organelles, and we don't see an R13B localized to these mutant organelles. So we wanted to use that knowledge to try and understand what are the molecular consequences to the bile duct cells, the cholangiocytes, after loss of the cilia. So to do this, we used a WDR35 flox transgenic mouse and crossed this in with a keratin 19 Cree that can be tamoxifen inducible. The idea being that we can specifically lose cilia in the bile ducts of the liver in these mice. And when we look at these animals 12 months after giving tamoxifen, we find the presence of these cysts all throughout the liver. In the normal control mice, we do see that there are these small bile duct cells that are hard to see probably on your screen. But at 12 months after tamoxifen dosing in the mutant animals, we see the presence of these large cysts. And when we compare the number of the cysts in the mutant animals compared to the number of normal bile ducts, in the wild-type mice, we see there's a significant increase. And these cysts are much larger in size as well. When we analyze the blood sera from these animals, we can see that there is increased levels of alkaline phosphatase, which is a marker of cholestatic disease. So we think the formation of cysts in this model does affect how these normal bile ducts function. And just to confirm that this mouse model is due to cilia loss, we stain for cilia, staining for the IFTB component, IFT888 in cyan and R13B in yellow. We can see through this cross-section in a healthy mouse the presence of these dual-positive cilia. And in our mutant animals, we see these rudimentary organelles, these puncta on top of the cells. We do sometimes still see R13B localized with these, with the IFT88, but the majority of these cells are R13B negative, as shown on the right side of that graph. So like I said, we were interested in understanding how does such a tiny organelle underpin the formation of this highly organized arborized structure within the liver. So you see you have this nice tree-like structure, but in the polycystic mice, we've seen the formation of these polycystic lesions branching off the main biliary network. And we have regions where they are dilated and other regions where they pinch in and form these septa, and we think this is what gives rise to new cysts over time. So like I said, we're interested in understanding what are the molecular signals that underpin cyst development. So we took our mice that were 12 months after tamoxifen dosing. We isolated the cystic epithelial cells specifically and the bile ducts of the normal mice. And we can see that by doing single-cell RNA sequencing, we have the emergence of this new transcriptomic cluster that is unique to the mutant animals shown in this peach color. And when we do differential gene expression analysis, we find that there are many different pathways that are altered. In particular, we see the presence of new pathways involved in cell adhesion, matrix reorganization, and cytoskeletal remodeling. And when we look at this a bit further, we find that there is expression of many different matrix proteins in the cystic cells. that are significantly upregulated compared to the normal wild-type mice. This includes laminins, fibronectin, and also collagens that I'm not showing here. And when we quantify this at the protein level, we find that these cysts are enriched in these matrix genes and compared to the wild-type controls. So we were quite interested by the fact that these cystic cholangiocytes are able to remodel the matrix around them. And we wanted to know if that was true in the human disease as well. So we recently did a spatial transcriptomics experiment using the genomics platform by NanoString, where we took FFPE tissues from patient tissues where we know the mutations of the genes, whether that be mutations associated with ADPKD or isolated polycystic liver disease. And we stained these tissues with pancytokeratin to demarcate our epithelial cells and CD45 to demarcate our immune cells. We also incubate these slides with oligoprobes, probes that can bind to the mRNA in situ. These can be cleaved by UV, and then we can then demarcate regions of interest and specifically extract the probes that are bound in situ and sequence them. And here's an example of one of these tissues that we looked at. And this is a patient tissue with PKD1 mutation. So we have this big hepatic cyst in the center, and we can see that there's cystic epithelial on either side and lots of stroma in between. And when we analyze the gene expression of these different cell compartments, we find that matrix reorganization, remodeling, is in the top hits for both the epithelial and immune compartments. We also analyzed the fibroblasts as well, and we could see that there was matrix remodeling there as well, but we thought that was less surprising compared to the other compartments. So we're curious to understand what is driving the expression of these matrix genes in the cystic cells. And we found that there was a strong expression of TGF-beta signaling. So we see expression of TGF-beta ligands, receptors, and downstream effectors. And when we look at this at the protein level, we see that there is little to no expression of TGF-beta receptor 2 in the normal bile duct, which is shown at the top here in magenta, but we see strong expression on the membranes of our cystic cells in the bottom panel. And to prove we have pathway activation, we see that there is little SMAD4 expression and little to no expression of phospho-SMAD3, which is active SMAD3. Whereas in the normal bile ducts, but in the cystic cells, we see that there is strong nuclear localization of SMAD4, and we have phospho-SMAD3 overlapping with that in the nucleus. So we can see that TGF-beta SMAD3 signaling is active in these cystic epithelial cells. Again, taking us to the human, we find the same signature, which is perhaps not so surprising in a chronic disease setting. But when we look at treating cholangiocyte cell lines from humans with TGF-beta 1, we found that they show expression of all the matrix genes that we see in our mouse model, as well as integrins, which are the predominant family for receiving matrix in mammalian cells. So to understand the essential nature of this pathway in vivo, we treated our animals with either vehicle or CYS3, which is a SMAD3 inhibitor. And we gave this to mice at 6 months and treated for 3 weeks. And we could see no significant change in the number of cysts in the treated group, but we see that these cysts generally are much smaller in size compared to the vehicle, or they're significantly smaller in size. And when we look at the blood sera, again, we see that there's a reduction in alkaline phosphatase, indicating that we're able to prevent cholestasis from progressing in this model. And when we look at fibronectin surrounding these matrix, we can see that we have a molecular readout. We see reduction of the fibronectin around these cysts compared to vehicle treatment. So just to summarize what I've shown so far, we found that loss of WDR35 results in loss of primary cilia in the bile ducts. This drives a cell-autonomous TGF-beta signaling pathway. And this allows for SMAD3-dependent transcription of matrix genes and integrin genes. And we were then curious to understand, is it the matrix and the integrin that are interacting? Are these the top hits that are new interactors within these cells? So using our single-cell RNA sequencing data set, we did ligand receptor prediction analysis, and we were interested in analyzing what were the new interactors in the cystic cells. And we could see that the majority of interactions in the new population on the right do involve the matrix and integrins, But we were particularly interested in integrin alpha-2, which is shown at the bottom of that wheel. Because when we look at this at the UMAP level, we see that integrin alpha-2 is specifically expressed in the mutant cluster, if you remember that as the bottom cluster. And again, when we validate this at the protein level, we find that there is no expression of integrin alpha-2 in the normal bile ducts, but we see strong expression in the cystic cells. And the alpha integrins have to interact with the beta integrins to form functional heterodimers. And when we look at integrin beta-1, both here at the RNA level, but also at the protein level, we find that there is strong expression across both normal epithelium and the cystic epithelium, as you'd expect. As well as seeing changes in the integrins, we also see that there's a general increase in the expression of downstream signaling effectors of integrin signaling. So that includes the genes that encode kindlin-1, integrin-linked kinase, and talin-1. And these all lie upstream of changes to the actin cytoskeleton. And when we look for phospho-myosin light chain 2 as a marker of active remodeling, we find that there is very little expression in the normal bile ducts, but we do see strong expression in the cystic cells again. And on the right was plotting intensity, and we can see that it appears to be more apically localized. And when we plot intensity as we travel from the basement membrane into the lumen of the bile ducts, shown in gray, or from the basement membrane into the lumen of the cysts, shown in yellow, we find there is this jump intensity. So we see an increase in active remodeling in these cells. This is also accompanied by changes in the size of cells, so the width of cells, and it gives us that characteristic flat shape that we see in our cystic tissues as well. So we were quite interested in integrin alpha-2, particularly because it seemed to be uniquely expressed in our in the cystic cells, but not in wild type. And we thought about how we could test this. So we've already heard about some kidney organoids today, and the kidney organoids are very nice, they're very sophisticated, but liver organoids are not as sophisticated. What we essentially do is we isolate a liver, we digest the parenchyma, and put the bile ducts in Matrigel, and we find that our organoids, instead of growing like tubular structures, I would expect, they just grow like big spheroids off the ends of these branches. That's what this looks like. We have a normal branch on the left, and we can see after 72 hours, it dilates and expands. And this is accompanied by an increase in the expression of integrin alpha-2 after 72 hours in culture. So if we treat with an inhibitor of integrin alpha-2 beta-1 called TCI15, we find that we're able to limit how big these organoids can grow in size, how much they can dilate, and that's plotted on the right. So again, what about the human tissue? When we look at non-cystic human liver controls, we find that, again, there is no expression of integrin alpha-2 on these bile ducts. But when we look at samples of polycystic liver that's come from patients, regardless of the underlying mutation, whether that be a mutation in the polycystins, or whether that be a mutation in the genes that are involved with isolated polycystic liver disease, we find that integrin alpha-2 is expressed. In addition to that, we find that when we treat the non-cystic human cell lines with TCI15, that inhibitor of integrin alpha-2 beta-1, we have no change on proliferation, which is shown on the left graph. But on the right, where we have cell lines that have been derived from a patient that had AD-PKD, had a mutation in GANAP, or a patient that had isolated polycystic liver disease and had this mutation in PRKCSH, we find that we're specifically able to affect proliferation in those cystic cell lines, but not in the non-cystic human cell lines. So what does this mean in vivo? So we first did a genetic study where we could specifically delete integrin alpha-2 in our same mouse model where we can model cystogenesis in the liver. Again, we gave these animals tamoxifen, and this time left them for 6 months. On the left, I'm showing you the normal control, where we have WDL35 and integrin alpha-2 both intact, and we can see these normal bile ducts around the portal vasculature. These are our 6-month time points where we can see cysts present in these livers in the absence of WDL35, but alpha-2 is still intact. However, when we also delete integrin alpha-2 at the same time point as we delete WDL35, we find that the majority of these livers phenocopy our wild-type controls. So by deleting integrin alpha-2 at the same time point as deleting cilia and driving the cystogenesis process, we can prevent cystogenesis happening. And that's just shown in this graph here, where we see this third group lines more closely with our wild type. But what about established disease? So this time we left our animals for 6 months and again treated for 3 weeks with this alpha-2 beta-1 inhibitor TSI-15. And here we find that we have actually a slight increase in the size of these cysts. It's significant, but it's only a slight change. But more interestingly, we find that by treating these animals with alpha-2 beta-1 inhibitor, we are able to reduce the formation of new cysts over time. So just to summarize what I've shown you, we find that loss of WDR35 or primary cilia drives a cell-autonomous TGF-beta signaling loop. This allows for the expression of the matrix genes and integrin genes. And if we inhibit SMAD3 signaling, we found that there was a decrease in the size of these cysts, so they don't dilate as much. So we think that the ability to form a new matrix allows for cysts to expand. The new matrix and the integrins interact with each other to drive active remodeling. And again, when we inhibit this in established disease pharmacologically, we are able to limit the number of cysts that form over time. However, if we delete integrin alpha-2 at the same time point as we delete cilia, we find that we're able to almost prevent cystogenesis in these mice.
5 minutes remaining.
I'm nearly finished, so that's quite good. So just to summarize everything, we find that SMAD3 and integrin alpha-2 serve as potential therapeutic targets in human polycystic liver disease. And I'd just like to thank Luke Bolter and Pleasantine Mill, who have been my mentors throughout this project and continue to be so, as well as everyone else who has contributed to this work, which involves a lot of people from across Europe. And I'd also like to thank funders and Early. So thank you.
All right. Thank you very much, Scott. It's a wonderful talk. And then I think we're going to move on to the Q&Z session. So I think I'm going to start with the first question. Can you hear me, Scott?
Yes.
Yes. So you showed that blocked ITGA2 genes, like in the liver cystic models, that decrease the cyst severity. I was wondering if you can also think about overexpressed that proteins can sufficient induce the liver cyst formation, because your wild type in vitro model seems showing that even the wild type cells have increase of that can induce the cyst formation. Do you have a thoughts on that?
My gut feeling would be that it would not induce cystogenesis. I think what we really need is the expression of integrin alpha-2 and the matrix, this new matrix that is made. So we find that there's a lot more of the collagen, laminin, and fibronectin that I spoke about, but we also see changes in MMPs and other molecules that are known to modulate the extracellular matrix. I feel like integrin alpha-2 alone would not be sufficient to drive cystogenesis, but we haven't done that experiment.
Okay, all right. So you got a couple questions. I'm going to rate that. From Dr. Fan Chen, and he was wondering, could you comment on whether duct plate malfunctions may be involving the cholangiocytes cyst formations in your mouse model and WDR35 patients?
Is that ductal plate malformation you said?
Sorry, can you say that again? Yes, the ductal plate malfunction. Yeah.
Yeah, it's a good question. So I think, if this was a patient that presented with a germline mutation in WDR35 or any other ciliopathies, it probably would be a ductal plate malformation, and perhaps recapitulate more what we see of the livers with patients with autosomal recessive polycystic kidney disease. However, I think because of this is obviously a model where we have induced loss of cilia in an adult mouse, essentially, P28. I think it is cystic, but I think it has probably both. It has features of both the dominant disease and what we see in recessive cholangiopathies or ciliopathies. And the integrin alpha-2 that we found, that is expressed on the patient tissue across different mutations, as I showed. So I think it's the model's perhaps somewhere in between. I'll say that.
Okay. So the next question is from Dr. Jing Zhou, and she was wondering, did you look at other integrins?
We didn't focus on them specifically, but if you saw my slide, there was the wheel of novel interactions that were predicted. So we do see other integrins like integrin alpha-3. We also see integrin alpha-V and beta-6, which is classically known as driving the activation of TGF-beta signaling as well, which complements our work. So we do see other integrins, but we didn't focus on them for experiments.
Okay. So next questions from Martin Prince. Is there a reason why cysts rise from the intrahepatic ducts, but not from the common hepatic duct?
It's a good question. I think in the liver field, we're quite familiar with the fact that we have these liver progenitor cells that typically reside in the more proximal tips of the biliary network, so right within the liver. So I imagine in our case we're seeing more cysts in that model, and actually in the mouse tissue we found that a lot of the cysts are around the edge of the liver lobe, where you'd find those small tips of the biliary network. So I think it could be something to do with the fact that they have more, I don't want to say stem-like potential, but they are more, these are the cells that respond to injury in the adult liver. So I think it could be reflected by that. That could explain why we find it more intrahepatic cysts.
Okay, so the last question is from Dr. Zitterman, and he was wondering, what are your thoughts on your data compared to data from the kidney showing that overexpression of TGF-beta restricts the cyst progression, or the data showing that the knockoff the ALK5 has no impact on cyst progression?
Yeah, I don't know. I mean, TGF-beta is one of those funny pathways where, from cancer, we're aware of the fact that it can have anti-tumor effects at the start of disease and then pro-tumorigenic effects, in chronic stages of disease. I don't know if that explains it for cysts as well, potentially. I should also say that, we see this canonical TGF-beta signaling pathway active, which feeds into the ALK5 part of the question, but we do also see non-canonical signatures. So we see BMP ligands expressed as well. So it could be that, we're able to impinge on this part of the disease when it's established, but I don't know. It could be a whole myriad of things. That's a good question.
All right. Thank you very much, Scott. And I'll turn back to the Francisco.
Great talk. Thank you so much. So I will introduce the next speaker, that is Dr. Ivana Guo. She's an assistant professor in the Department of Cell and Molecular Physiology at Loyola, University of Chicago, and she's interested in cardiovascular manifestation of ADPKD. I read many of her papers showing how alteration in polycystin 2 can drive cardiovascular alterations in cardiomyocyte directly. So the title of her talk is Sensing Stretch, Cardiac Polycystin, Natriuretic Peptides, and Hypertension. So thank you so much, Dr. Guo, for you.
Thank you so much for that kind introduction. You can all see my slides.
You're in presenter view. We need the presentation mode. Just swap displays up there. Yeah, that's good.
Yeah, that's good.
Thanks again. So we saw this slide presented by Dr. Somlo yesterday, which talks about the progression of polycystic kidney disease. What I'm going to focus on today is this lower part here, where we see that hypertension is one of the earliest symptoms seen in PKD patients. And if you talk to any patients, oftentimes you'll hear anecdotal stories that they develop hypertension in their teens, oftentimes decades before they get a formal diagnosis of PKD. And you can also see that this hypertension occurs before the expansion of the kidneys that is used to classify with the MIC system. So the other thing about cardiovascular or extrarenal manifestations is that in the era of renal replacement therapies, and by that I mean dialysis, as well as kidney transplant, cardiovascular manifestations are the main cause of mortality in PKD patients. So our lab works towards understanding the roles of cardiac polycystins and going forwards, the crosstalk between organs in the contribution of this pleiotropic disease. So what are the sources of hypertension in polycystic kidney disease? There have been many studies going back decades since the discovery of the polycystins that have described that their hypertension seems to be driven by the cysts compressing upon the renal vasculature. Following that renal vascular compression, there is an upregulation in renin, followed by the downstream cascade, including the upregulation of renin, leading to aldosterone, and as well angiotensin. Now, most of you will know that aldosterone and angiotensin are going to be promoting the retention of salt. And indeed, the angiotensin pathway is definitely the target of blockade by ACE inhibitors and ARBs. So in addition to all of the salt retention that can happen, angiotensin 2 also elicits vascular effects, as you can see here in the slide. Now, I want to draw your attention that when we have hypertension, that then can feed forward and ultimately the ability of the kidney to regulate the amount of blood flow is compromised, and that then further exacerbates the renal disease. But as I mentioned earlier in my introductory slide, hypertension is an early symptom seen in PKD patients, and it is seen oftentimes before we get this, the cysts that are going to be pressing upon the renal vasculature. So what then are possible non-renal sources of hypertension? Indeed, we see that in patients with ADPKD that they have compromised vasculatures, and this includes an impaired endothelial function, so that they have decreased amounts of nitric oxide being produced. And nitric oxide, for those that don't know, is a vasoreactant. However, my talk today is going to focus more on this impaired natriuretic peptide production from the heart. So how does the heart contribute to overall blood pressure and also potentially the manifestations that we see in hypertension in the PKD context? Now, the heart, as we all know, is the ability, pumps blood around our bodies, and when there's a volume overload, that's sensed by the heart as this distension, and that releases what is known as natriuretic peptides, commonly known as ANP and BNP for atrial natriuretic peptide, or BNP for brain natriuretic or B-type natriuretic peptide. I won't go through all the mechanisms, but what I want to impress upon you is that they actually counteract a lot of the renin signaling pathways. The very name, the N standing for natriuresis, means that you get salt wasting, and collectively you get decreased blood pressure, because one of the targets of ANP and BNP are the NPR 1 and 2, which when activated either on the cardiac cells or in the vasculature, and indeed also in the kidney, result in cyclic GMP signaling, which enables cells to relax. So you can see that there is definitely an interplay here between cardiac cells being able to sense stretch and then the downstream effects on the kidney and blood pressure. So in the study that I'm going to share with you today, we wanted to test if loss of cardiac polycystins contribute to the high rates of cardiovascular dysfunction that is seen in PKD patients, and also does it contribute to hypertension. Now, the way that we have gone about this in my laboratory over the past few years is actually to use a cardiac-specific knockout mouse of polycystin 2. So if you look back into the earlier studies of PKD, you would see that the heart actually expresses quite a significant amount of polycystin 1 and 2. Now, in our mouse model, we are not actually touching the kidneys. In other words, this is not a cystic mouse. What we do is we wait until the mice are around 6 weeks of age, and then we induce the knockout, in this case, of polycystin 2 with tamoxifen. And the Cree driver here is alpha MHC mercury mer, which means that there is restriction of the expression of Cree to only the cardiomyocytes. And you can see that here, the polycystin 2 adopts a very punctate staining throughout the cardiomyocytes. And this is really lost when you look at in comparison to the knockout. I want to emphasize to those that aren't so familiar with cardiomyocytes that unlike the previous talk, which talked about the cholangiocytes, or other talks that we've heard about the kidney epithelial cells, cardiomyocytes really do not have any cilia. The only ciliated cardiomyocytes are those that are endocardial and facing into the chamber. So for the purposes of this talk, I am talking about polycystin here that is actually expressed within what is actually going to be the endoplasmic reticulum, or actually the sarcoplasmic reticulum within cardiomyocytes. You can see, and you can appreciate hopefully by the histology, that there is really no outward remodeling of the heart here. And we also do echo analysis to look at cardiac function. It's showing you the systolic function as measured by ejection fraction. And between these data, you can see that there is really no objective systolic dysfunction in these mice. We also implanted our mice with telemetry devices to measure their blood pressure. So these are implanted into the carotid artery. And so that allows for 24-hour monitoring day and night. Now, I should also note, as I said earlier, that these mice are non-cystic, and so I just want to show you this slide to demonstrate indeed these mice are not cystic at all. When we looked at our telemetry data, what we found was somewhat surprising, given that we did not see a change in their cardiac function, that these mice were actually hypertensive. And the age at which these data were collected was about 4 to 5 months post-tamoxifen induction. So this hypertension was seen in both sexes. And for anyone that studied hypertension in a mouse model, you would appreciate that seeing hypertension in a mouse that has a C57BL6 background is going to be somewhat surprising. So we were actually quite taken aback by the robustness of this phenotype here. We see both reductions, we see both increases in the systolic as well as diastolic pressures, again, shown here with the S and the D. And heart rates were not changed. So I mentioned that there was this connection between natriuretic peptides and salt wasting and hypertension. But how then does polycystin 2, or the loss of polycystin 2, affect this whole natriuretic peptide signaling pathway? Well, the reason why we wanted to look at the natriuretic peptide signaling pathway is because it's known that mutations to natriuretic peptides in humans can either lead to exacerbated hypertension when not enough natriuretic peptides are made, or protection against hypertension when the natriuretic peptides are not degraded. So the way that natriuretic peptides work is that they are thought to sense the distension in stretch. So cardiomyocytes on a beat-to-beat basis, they undergo contraction. But when they contract, you also get the potential stretch between cells themselves, right? And so the cells, the cardiomyocytes, are able to distinguish between what is physiologic stretch and pathophysiologic stretch. And so what happens with pathophysiologic stretch is that it gives this excessive stretch that produces natriuretic peptides. And this natriuretic peptide production results in the upregulation of NPs at the molecular level, as well as the release of natriuretic peptides from the left ventricle as well as the atrias. And as I said, those natriuretic peptides results in vasodilation and natriuresis, and they can be degraded by neprilysin. Now, where can polycystin 2 potentially link into this? The release as well as the upregulation of natriuretic peptides is calcium dependent. The calcium here is not due to the beat-to-beat calcium that is seen, so it seems to be independent from that. But the source of this calcium so far has not actually been well understood. Now, we posited then, because we know that polycystin 2 can act as a non-selective cation channel, that perhaps, and because of its virtue being located to the sarcoplasmic or endoplasmic reticulum, it could function as a calcium source here. So it could function there. So first of all, we wanted to look and characterize what was going on with the mRNA, as well as all these pathways, to see if polycystin 2 was indeed participating in natriuretic peptide signaling. And what we saw is that in the left ventricle, we saw there was a downregulation, as you can appreciate here, of NPPA, which encodes for natriuretic peptide ANP, as well as B, but this was reversed in the males, though with NPPB, we do see this downregulation. At the protein level, we saw that both sexes, there was a downregulation of BNP. So despite the fact that there was an upregulation in the mRNA, this did not translate to the protein level. Additionally, and this is important, given that the mice here that we're talking about are hypertensive, we did not see an upregulation in the natriuretic peptide signaling circulating. And this really, for us, made us think for a moment, because what we've got is an animal which is hypertensive. It's not releasing natriuretic peptides, and it's not responding to the fact that the animal is hypertensive to release more. And all of this seems to be driven by the loss of polycystin 2 in the heart. Moreover, we checked the circulating renin levels, and if anything, again, as I said, that the kidneys are intact, we saw that there was no change in circulating renin. Now, some might ask, where does this circulating AMP and BMP come from in these animals? And it comes from other sources. So natriuretic peptides aren't just made by the heart. They can be produced by the kidneys. They can also be produced by the brain. So it's not surprising to see some levels of natriuretic peptides in our animals. Given this phenotype, we also looked carefully to see if there was any change in salt handling within our mice. And here you see that there's no change in body weight. We also checked for the amount of food that our mice consumed, and we saw that there was no change over 24 hours. However, we did notice that our females drank less water, which seemed to be consistent with trying to ensure that the overall homeostasis within the mouse was remained consistent. Urine osmolality was unchanged, and the fractional excretion of sodium was unchanged. So going back again to the cardiomyocyte phenotype that we saw, which is that the natriuretic peptides did not seem to be produced in our cardiac-specific knockout mice. So what's going on? I've already shown you that there was downregulation of the natriuretic peptides in our knockout animal. How about the other signaling pathways? Well, we looked at the chromogranin as well as NF-kappa B. And chromogranin actually allows for, it's one of the scaffolding proteins that enables for the exocytosis and the vesicles to form. And NF-kappa B also lies downstream of chromogranin B, but it also is a calcium-dependent transcription factor that, when goes to the nucleus, allows for the production of natriuretic peptide B. And so what you can see in both males and females, that both of these genes are downregulated. And these data are taken from the left ventricle. Moreover, when we looked at the processing side, so anyone who is familiar with the natriuretic peptides would know that natriuretic peptides aren't just made as an active sequence. They actually undergo a number of cleavage events. And one of the secretases that is used is corin, as well as PCSK6, which regulates corin itself. What we found is that PCSK6 is downregulated in both male and female mice, as well as corin having a trend downwards. So putting all this together, what we see is that there is a downregulation of the mRNA in the cardiomyocytes. There's downregulation of natriuretic peptide processing, as well as components involved in the actual vesicle formation. But how about this calcium part? Where does polycystin 2 again fit in there? So to address that question, what we did was we crossed our mice with a GCamp reporter, TD tomato, which is known as salsa mice, and we imaged the right atria. And the reason for this was both ease of imaging, but also because the atria can easily distend, and it's actually the source for the A for atria in atrial natriuretic peptides, a source of natriuretic peptide production. So with this preparation, what we can then do is GCamp is a calcium indicator. So what we see is that we can measure the amount of calcium on a beat to beat level. Now, these hearts are spontaneously beating. In other words, this is relying on the sinoatrial node. So there is no exogenous stimulation going on here. As a proof of concept to ensure that our system was intact, what we did was we stimulated the cells with isoproternal, which is a beta receptor agonist. And you can see the elevation in calcium shown here in the orange trace. So what you're seeing here is the calcium transient normalized to the TD tomato to ensure that there are no motion artifacts. And what I think you can appreciate in the quantified data is that both in the control and in the knockout, we see this upregulation of calcium with agonist stimulation. Now, some of you may be scratching your heads because a lot of the PKD field says there's always a downregulation of calcium signaling. I will say that in the cardiomyocytes, we've got this interaction with the renin receptor. And so what we're seeing is that the polycystin 2 actually has an opposite effect on the renin receptor compared to what we see in renal epithelial cells. But how do we stimulate an excessive stretch in these preparations? So to stimulate an excessive stretch, what we did was we actually infused in a hypoosmotic stimuli. So the argument being here is that when we infuse into our preparation hypoosmotic stimuli, going from 300 milliosms, and now we were decreasing the amount of Na and Cl, but keeping all ions constant, we would actually enable the cells to actually expand and swell because they're going to counteract that osmotic stretch. And so what you can see and appreciate here is that when we do this in the control, we actually get an upregulation or an increase in our calcium signaling. However, in our knockout, this is not seen at all. So there is no additional calcium release that is seen in our knockout with this hypoosmotic stretch. So that's in the atria, but that's to do with the calcium. Does this actually translate to actual vesicles? To address this, we used electron microscopy, and here is the Cree control left atria, and hopefully you can appreciate the presence of these vesicles there that really are not very rarely seen in the knockout. Now, those data are all taken from the mice. So we wanted to address if this could actually be seen in a human preparation. And to do that, what we did was we collaborated with a colleague of mine, Dave Bearfield, and he took human iPSCs and derived them into cardiomyocytes. Now, as they mature, they beat. We then seeded them into these myopods, which allows for alignment of the cardiomyocytes, and then infused in a lentivirus containing either a polycystin 2 guide sequence or a non-template control. We then put them into a force transducer to measure the amount of twitch, or in other words, contraction that was happening with forced pacing. And the reason for this is because when the previous experiment using that spontaneous calcium that comes from the sinoatrial node, it's quite possible that there could be other factors that are being affected there. So this allowed us to control the amount of signal that we are actually stimulating the cells with. So with this, we can measure a force of the contraction and use that as a readout. And you can see 4 lines that nicely overlay. And this is important because what it tells us is that there is no change with the force generated in the knockout or the knockdown cells compared to the control cells, either in hyper or hypo conditions or under normal osmotic conditions. And you can see here in this preparation, what we're doing is we're actually stretching the preparation to try and induce a stretch of the cardiomyocytes. when we took these preparations, despite the fact that they had the same amount of force that was generated, and we looked at the amount of ANP that was produced, what we saw that there was a marked decrease in ANP puncture size in the knockouts.
5 minutes remaining.
So that's all well and good in the human iPSCs, but can we restore this function? So turning to a myoblast cell model, what we did was we checked for natriuretic peptide production, and both at the mRNA level as well as the protein level. And so here you can see that in the knockout cells, we see a downregulation in NPPA. When we express two different mutant forms of polycystin 2, either the D511V, which is in the voltage sensing domain, or a truncation, we get variable results. The D511V restores expression similar to the polycystin 2 add back, but the 703, which is the truncation, does not. When we then turned to a assay to look at puncture formation, and here we're using ANP fused to an emerald, so we can see the puncture here forming there in our knockout, in our control cells, and in the polycystin 2 add back cells, but the puncture is not seen in the knockout or the mutant D511V. So functional polycystin 2 is required for this NP puncture formation. What about polycystin 1? We see the same thing with our ANP emerald assay. In other words, we don't see puncture formation. And in the published data, we see that polycystin 1 actually adopts this location to the intercalated discs, and it nicely lines up with Z01. This is using the E4 antibody. And so what we think is that polycystin 1 here is located at the junction of the cells, setting the stretch. So together, what we see is that we think polycystin 1 is located at the junction of the cells. It perhaps could interact with polycystin 2, but together the two interact to regulate multiple aspects of the natriuretic peptide signaling pathway, going from transcription all the way to the production of the vesicles. Ultimately, we end up with hypertension due to the downregulation of ANP and BNP being able to be released. So in summary, hypertension is an early and common manifestation in PKD. Both polycystin 1 and polycystin 2 are present in cardiomyocytes. In the polycystin 2 knockout mice, its loss contributes to hypertension due to the loss of an NP signaling pathways, and we see impaired puncture in both polycystin 1 and 2 models. Our future directions are to look at the effects of Entresto. Some of you may be familiar with Entresto. It's actually coming off the patent in a few months. It's actually a combination of sacubitril and valsartan. So sacubitril is a neprilysin inhibitor, and it has been used in Entresto form to treat heart failure, as well as more recently hypertension, and seen some reports in chronic kidney disease. It has not, to my knowledge, been tested in PKD patients. So we're excited to test that out in our animal models. And we're also doing crosstalk studies between the kidney as well as the heart to see how natriuretic peptides and the renin signaling pathway that happens and is elevated with this crosstalk together and perhaps exacerbates cystogenesis. So with that, I would like to thank Carla Marquez Nogures, as well as Brandon Elliott from my lab, who spearheaded a lot of the studies that I presented to you today. You can see our funding sources, and thank you all for your attention.
Thank you so much for this excellent talk. So we are going to start the Q&A. And there is one question in the chat. But before I ask for that, I wanted to ask a question. So did you study long-term changes in hypertension or cardiac remodeling in your mouse model?
Yeah, so that's an excellent question. So in the data I presented today, that was 4 months after its knockout. In previous studies, we have studied the knockout up to 9 to 10 months after post knockout. We've also got ongoing studies with TAC, pressure overload, as well as myocardial infarction.
And do you see any progressive increase in hypertension, or is kind of stay at the same level?
Yeah, so in the months that we measured it, about 6 weeks, we did not see it being progressed.
Okay, good. So I will continue with the chat. So great talk. This is from Michael Godgen. A great talk and very convincing data on the physiological role of cardiac PC2 in the control of blood pressure. Do you think that this mechanism can account for high blood pressure in ADPKD patient where the vast majority of cardiac myocyte is heterozygous for mutant PKD2? Have you checked whether you see an effect of AMP and blood pressure in heterozygous mice?
Yeah, no, that's a fantastic question. Thanks for that. Yeah, so in prior studies, and actually one of the reasons for looking into the natriuretic peptide pathway in our cardiac specific knockout was we took Dr. Somlo's heterozygous polycystin 2 whole body heterozygous mouse when I was a postdoc, and we saw that with angiotensin as well as isopaternal stimulation that should result in cardiac remodeling, the heterozygous polycystin 2 mice had blunted AMP and BMP levels. So although we didn't test the blood pressure in those mice, we did see, at least in the left ventricle, that there was a reduction in AMP and BMP.
There is another question in the chat from Dr. Kian. Do kidney-specific PC1 or PC2 cardiomyocyte-specific knockout causing cystic kidney formation have hypertension?
Yeah, so that's
No.
actually a great question. And so there's been some smatterings of that. And I would say that Tucker's work has shown that there is an upregulation in the renin signaling pathway. And so that seems to be one of the main drivers from the kidney side of things. There's also work that has suggested out of Utah that in the pre-cystic polycystin 1 knockout mouse, those mice are actually hypotensive and not hypertensive. I think more specific studies need to be done with just the cysts, though.
So the next question is from Dr. Cockerell. A very interesting talk. Do you observe any left ventricular hypertrophy in these animals in response to hypertension? Would it be possible that they are protected to stretch-induced hypertrophy?
Yeah, so that actually is what we do indeed find. If anything, there is a diastolic dysfunction, not a systolic dysfunction. Regarding the hypertrophy, we actually don't see as much remodeling, and in our TAC data, which is still working on that, we actually see that the polycystin 2 cardiac knockout mice seem to be protected from that excessive stretch.
So the next question is from Dr. Sussman. I believe these mice also get cardiac fibrosis. Can you comment on how that could relate to this data?
Yeah, thank you for that question. So we looked carefully for fibrosis. I would say that in comparison to some of the other cardiac models that have been published so far, our model is induced at adulthood, 6 to 7 weeks post-birth. Other models which have knocked out either polycystin 1 and polycystin 2 have been earlier than that time point. So yes, in those mice, you do see fibrosis. We used alpha SMA and did not see a difference in fibrosis between our control mice as well as our knockout mice.
Thank you so much. Any more questions? We still have a little minute, like a couple of seconds. Okay, if there are no more questions, I will give the word to Dr. Song that will introduce the next speaker. Thank you.
Thank you. It is my great pleasure to introduce the next speaker, Dr. Maureen Barr. Dr. Barr is the distinguished professor of genetics at Rutgers University. The Barr Lab uses the C. elegans to study fundamental questions in the cilia biology and to understand a human genetic disease. And the Barr Lab pioneered the use of the C. elegans to study extracellular vesicle biology. And welcome, Dr. Barr, and looking for your talk.
Hi, can you hear me okay?
Yes.
Great, okay. Hello from New Jersey. I am way beyond the kidney. I want to thank Terry and Brad for inviting me. Today I'm going to talk about something we haven't heard about, and that is the ability of cilia to produce extracellular vesicles. So here is shown one of our favorite organelles, the cilium. We know that it functions as a sensory antenna. It also has the ability to release extracellular vesicles that could be involved in the signaling function. And that's what I'm here to talk to you about today. So the outline of my talk is I'm going to give you a little bit of history, talk about things that happened a quarter of a century ago. I think this is important for the trainees to appreciate. Then I'll talk to you about ciliary extracellular vesicles and how the polycystins recruit cargo to ciliary extracellular vesicles. And time permitting, I'll talk to you about how we're using this tiny worm called C. elegans as a model for studying pathogenic mutations and variants of uncertain significance. So going way, way back, why I'm here is as a postdoc, I was really interested in the question of how an organism senses and responds to its environment. And I chose to use this tiny worm, C. elegans, because it has a really simple nervous system. The genome was entirely sequenced. Many of the genes in the worm have counterparts in the human. Most of the studies in this tiny worm were done in this modified female called the hermaphrodite. And the male was the neglected of the two sexes. He has an additional set of neurons that are important for mating behavior. So as a postdoc, I screened for genes that were important for mating behavior. This is an unbiased screen that brought me to the polycystic kidney disease genes. So what I found was that the C. elegans counterparts of PKD1 and PKD2, we call them LOVE1 and PKD2, act in the same genetic pathway. They have a similar structure. They localize to cilia. This was in 1999. Some of you probably weren't born yet. What we showed, here's the tail of the worm, and these arrows here are showing you LOVE1 and PKD2 localizing to cilia. And here's the head of the worm, and here's the nose. And here's LOVE1 and PKD2 localizing to cilia. The microscopy was terrible back then. I'm going to show you some much better images. So in this type of paper, I think we got 2 paragraphs to speculate on what we thought was going on. So I took a reread of our last paragraph and I wrote, LOVE1 might act in a sensory signaling pathway. And LOVE1, which is polycystin-1-like, may function as a molecular scaffold for other molecules. And what I'll talk to you about at the end of this presentation is that, in fact, LOVE1 does act as a molecular scaffold. It recruits cargo to ciliary extracellular vesicles. So this is an entirely new function for polycystin-1 that we're really excited about. Some more history. So that paper was published in September of 1999. This email did exist, but we got snail mail, this little envelope came with an index card from Joel Rosenbaum, who said, "Dear folks, you probably don't realize it, but you've just published on interflagellar transport protein." So Joel was the discoverer of interflagellar transport in Chlamydomonas, which is an algae. We've heard an awful lot about IFT 122, 140. This was all discovered in an algae. And he contacted us 1 month after this paper was published. And this is really the beginning of this incredible career that I've had. And because I'm the second to last talk, and the next session is going to be with trainees, I really wanted to talk about the power and importance of mentors, collaborators, colleagues, and supporters in my own career. So for trainees, it's important that you develop a robust network of mentors. And then for us PIs, it's really critical now to pay it forward and to support our trainees. So some ancient history. In 1998, I reached out to Greg Jamino and Fang Chan and asked for PKD1 and 2 cDNAs. As a consequence of that, Greg reached out to my postdoc advisor, Paul Sternberg, and said, "Hey, do you want to be on this program project with us?" And Paul's like, "You want to do that?" I'm like, "Yeah, sure." So we wrote this, it got funded. Paul generously, there were no K99 or 00s then. He let me take half of it to my own lab. At the time, I was at University of Wisconsin in Madison. This is April 2000. Johns Hopkins had a PKD Center meeting where I met Greg, Terry, Craig Montel, and Fang Chan. I visited Yale a month later. There was an ASN meeting where I met Steve Somlow. A few years later, I was invited to present at the PKD FASIB meeting where I met many PKD folks, including Lisa Gay Woodford and Brad Yoder. There was Cilia subgroup meeting, which is now like really a key part of ASCB, where I met many Cilia folks, including Greg Pizzer. I had early funding from the PKD Foundation to study genes important for PKD2 ciliary localization, and this provided the foundation for my lab. So I want to acknowledge the PKD Foundation for funding this basic research. And then in 2005, Peter Harris invited me to Mayo to give a talk where I met Chris Ward and Marie Hogan, and a few years later, they told me about the polycystins being shed in urinary extracellular vesicles. So I had all of these really instrumental contacts early in my career, and I've been fortunate to be funded by the NIDDK continuously for 24 years, and I've had amazing program officers, including Christine Marie Broken. So I want to thank everybody. Okay, so here we study C. elegans, polycystins. Here's the male, his head and his tail, the polycystin proteins. Here's a simple C. elegans sensory neuron. It has a cell body where there's ER, axon, dendrite. In worm cilia, in worm neurons, cilia are located at the distal ends of dendrites. You can see PKD2 in cilia in the head and in cilia in the tail. Here's LOVE1. It has the same structure as polycystin 1. It's also enormous. It's 3,281 amino acids. It has a GPS site that is important for its function and its cleavage. It has top domain. Here's PKD2. It's a trip channel. It also has top. Really, the surprise came in about 10 years ago now, when Jan Wang, my right-hand woman, discovered that the polycystins were shed from cilia in C. elegans. So here I'm showing you LOVE1 tagged by M. Scarlet and PKD2 tagged by GFP. These are endogenously tagged proteins. And here's the tail of the male. These are sort of like finger-like structures that are important for sensory function. And what you can see here is that you don't see green, you don't see pink. What you see is white because LOVE1 and PKD2 co-localize in cilia. And here you can see they're shed. They localize to the cilia base, the cilia shaft, the tip, and here you can see them being shed in extracellular vesicles. So the worm has a lot of advantages for studying the polycystins. So their structure is evolutionarily conserved. They act in the same genetic pathway. They localize to cilia and they localize to extracellular vesicles. This is all similar to what occurs with the human polycystins. We can visualize the polycystins in living animals in real time. We have functional assays for for PKD2 and LOVE1 with mating behaviors, genetic tractability, and the worm is super. We don't have to worry about any protocols. We can go from 1 worm to 300 worms in 3 days, so it's rapid and cost effective. So EVs. EV shedding is a superpower of the cilium. Here's our favorite organelle shedding EVs. This is work from Nick LaRusso's lab in the ARPKD mouse model. You can see in this fixed TM image, EVs accumulating or being shed from cilia. You can't tell the difference with the static image. Here's an SEM showing EVs associated with cilia. A more recent paper from Gaia Pagino is showing vesicles budding from MDCK 2 cells. And here's an earlier paper showing a mouse neuropil showing prominent with immunogold associated with EVs being shed from vesicles. So the polycystins localize on EVs, which are these really tiny vesicles that escape detection by light microscopy. We want to study the function of the polycystins on EVs. The huge challenge is that blood, sweat, tears, urine have EVs, and we don't know the source of these EVs. So there's high heterogeneity. A cell can make EVs by many, many different biogenic mechanisms. So what we're doing is really now taking full advantage of C. elegans. We used it for a decade and a half to study polycystin ciliary localization. Now we're all in on the fundamental biology of EVs. So today I'll talk to you about EV cargo sorting. I'll give a little bit of background about what we know about EV targeting and bioactivity, and then more recent work on EV omics. And I really want to point out that Jan Wang, as my right-hand woman, all of our early EV studies were done by her. And more recently, Ina Nikanarova joined the lab, who's an extremely accomplished biochemist, and she's done some amazing work on determining the function of polycystin carrying EVs. So here's our C. elegans animal. It's transparent. PKD2 is a conserved EV cargo. This is a cartoon of the ciliated neurons that express the PKD genes. Zooming in, here's that tail with the finger-like structures. The polycystins localized to cilia, and they're shed in these extracellular vesicles. Again, you've seen this. And we also showed by immunogold labeling that endogenous polycystin or LUV1 localizes to EVs. So we know that the polycystins are important for inter-animal communication. So this is our really convenient functional assay for polycystin function. So the polycystins are important for mating behaviors. We also showed that these male neurons that shed EVs, so when the male is mating with its partner, he transfers EVs to her cuticle or skin. So here's the male, if you picture, he's like sort of a footprint on the modified female, and he's depositing polycystin EVs on her cuticle. We don't know the significance of this yet, but we're really interested in understanding how and why this is occurring. So now, the more recent things that we've been working on, the goal is to identify the composition of the polycystin carrying ciliary EVs using C. elegans as a model system. Here, Ina made a cartoon of EVs in a worm and EVs in a human. So we're using C. elegans as a system to decipher molecular mechanisms of polycystin EV shedding. We want to develop new tools for the EV field for analysis with the goal. So we're not going to use C. elegans to find cures or treatments for ADPKD. What we want to do is use C. elegans as a discovery system. So, could we use C. elegans as a clue to how we could facilitate diagnostic approaches? So, for example, Chris Ward is trying to use EVs for diagnosing ADPKD, urinary EVs. We really would like to use C. elegans as a system to screen and identify potential targets for future therapies. So the challenge, as I said, is EVs are really small and they're heterogeneous. And the solution is that we expanded our C. elegans culture. So rapid and quick, we can do that pretty easily. We've developed methods to enrich the vesicles of interest. And really the main solution was Dr. Ina Nikanarova, who joined the lab with the goal of tackling this extremely challenging project. The work that I'm going to talk about was put on bioRxiv about a year ago. Title is Polycystins Recruit Cargo to Distinct Ciliary EV Subtypes. So the way Ina first started was to take advantage of our PKD2 GFP labeled EVs. So she did bulk culture of C. elegans, harvested extracellular vesicles through different steps of ultracentrifugation. She fractionated and then selected those fractions where she could see PKD2 GFP. And what she found was that there were 3,000 proteins in the C. elegans eviome, which should not have been a surprise to us because almost in humans, every cell in our body sheds EVs. The same is true for C. elegans. Many tissues in the worm make extracellular vesicles. So she found 3,000 cargo, and of that, she estimates that polycystin EVs are a very small percentage of EVs that are shed by the animal. So what we really want is to find these EVs that have just PKD2, and we want to see who are PKD2's best friends in these vesicles. And what she did was to develop this really clever strategy. It's sort of an indirect proximity labeling. So she goes through the same process, but now she has a second marker in this strain background. So here's PKD2, GFP on a vesicle. What she did was co-express a transgene that has turbo ID, which is proximity labeling enzyme, fused to M scarlet so she can see it. And then she uses a nanobody to GFP so that she can specifically target turbo to PKD2. And then if she wants to see PKD2 and EVs, she can isolate EVs. So this is showing you the strain that is expressing endogenous PKD2, endogenously tagged PKD2 GFP with turbo ID. Again, here's this male tail with those finger structures. Zooming in here, the inset, we can see PKD2 GFP EVs, and we can see turbo is targeted to those EVs. And here, if you look here, you see a lot of white EVs. So turbo is targeted to ciliary extracellular vesicles. And what she found was that instead of 3,000 proteins, she found a handful of cargo that was associated with PKD2. Because of the rapid and inexpensive nature of C. elegans, she tagged each of these cargo using CRISPR-Cas9 editing. She was able to knock out these proteins and to basically to demonstrate that these are bona fide cargo of polycystin carrying EVs. So she found LOVE1, not unexpectedly. She found a channel-like protein that has characteristics of a chloride channel. She found transmembrane lectins that we hypothesize are involved in adhesion and ligand binding. And then she found 2 tumor necrosis receptor associated factors, TRAF1, which has an E3 ligase domain, and TRAF2 that does not. These function as signaling adapters. So I'm going to just, for time, so show you only one cargo and give you the take-home message. So what she found was that the membrane lectins, the channel-like proteins, and the TRAFs all co-locate with PKD2 and LOVE1 in EVs. If she takes away LOVE1, so knock out LOVE1, what she finds is that PKD2 still gets targeted to EVs. In contrast, the TRAFs, the channel-like protein, the membrane lectins don't get targeted to ciliary EVs. So she concludes that LOVE1 is essential for loading of cargo to ciliary EVs. This is a brand new function for polycystin-1-like protein. And as an example, I'll show you what she found for TRAFs. So she did the same logic was applied to all the cargoes. So here, PKD2 is GFP tagged. The TRAF, one of the TRAFs, which is homologous to human TRAF6, is tagged with M scarlet. You can see TRAF1 locates to the cilia base, the shaft. PKD2 is there too. What you can see is not green, but you can see where they co-locate. So you can see co-localization of PKD2 at the, and TRAF1 at the base along the shaft. And here, if you look at the tip of the cilium, they're co-locating, and shed into the environment are PKD2 and TRAF1. When she knocks out LOVE1 or polycystin-1, TRAF1 and PKD2 still locate to the cilium. PKD2 in green is still shed in vesicles, albeit at lower levels, and she doesn't see TRAF1 being shed there. So again, LOVE1 is required for loading of TRAFs to ciliary EVs, but not PKD2. So Ina scoured the literature to see if there's any evidence for TRAFs being involved in PKD or in ciliopathies. And indeed, TRAF6 knockouts in mice display ciliopathy phenotypes. So mice have exencephaly, alopecia, shortening of long bones, teeth agenesis, and delayed pigmentation. TRAF7 functions in cilia of vertebrates, zebrafish, and xenopus. So here's wild type mucociliary epidermis. Here's the pronephros cilia from xenopus. And if you look at the TRAF7 knockouts, cilia are misformed. And in the xenopus, there are vesicles accumulating between the axoneme and the ciliary membrane. So it doesn't look like this is a weird worm thing. It looks like the TRAFs are functioning in invertebrate and mammalian cilia. So our model, so what I'm hoping that I've convinced you is that C. elegans is really powerful for dissection of EV signaling. And what I propose, or what we propose, is that the polycystins have cell autonomous function, so in cilia, in ER, but we also propose that they have non-autonomous function.
5
EV
minutes
function.
remaining.
Thanks, I'm almost done. And this is a harder thing to test in anything other than a worm. So I hope I can get people thinking about this. We heard about from Michael Kotkin that DNA, J11, mice have the cysts form in the proximal tubule. And the question is, well, what's going on in the distal? Is it possible that cilia in the proximal tubule are shedding EVs that are traveling to the distal tubule and affecting the distal tubule cell non-autonomously? I do not know. Here's our wild type cilium, LOVE1 and PKD2 are shed in vesicles along with associated cargo. We also have independent cargo that don't need the polycystins for their location. So in normal situations, we'd have polycystin 1 and 2 associated cargo and independent cargo. I showed you that if we knock out LOVE1, all of those other cargos are no longer associated, but vesicles are still made. They only carry PKD2 in this independent cargo. If we knock out PKD2, nobody leaves the ER. They all stay stuck in the ER, but vesicles are still made, and that independent cargo is on the vesicle. So the hypothesis is that not only can you have polycystins affecting the function of the cilium, you can also have polycystins affecting the signaling functions of EVs, and that those could have pathological consequences. So I will sort of say we're using C. elegans as a model for pathogenic mutations and variants of uncertain significance. We can mutate the GPS site and show that that destroys the function of LOVE1 polycystin 1. We have preliminary evidence showing a pathogenic mutation in the top domain of PKD2. The protein's still made, it doesn't localize to cilia or EVs anymore. And this is something we're just starting. We're really excited. If anyone here has a mutation in PKD1 or PKD2 and it's conserved in C. elegans and you want to see what it's doing, please reach out. We'd love to collaborate. And the reason again for that is C. elegans is great for polycystin research because their structure is evolutionarily conserved. They act in the same genetic pathway. The polycystins localize to cilia in a worm and in a human. They localize to EVs in a worm and in a human. We can image things in vivo. We have really straightforward assays. Genetic tractability is amazing, and everything is quick and easy. And then I just want to really give a shout out to my amazing collaborators. Jan has been leading EV biogenesis and bioactivity aspects. Ina has been taking the charge of EV omics. I only talked about proteins in EVs. She's also looking at RNAs in EVs. And I didn't have time to talk about Katie Jacobs' work, but she's looking at the communication between cilia, EVs, and ECM. And then finally, we've been generously supported by the NIDDK, the PKD Foundation, and Jan received a pilot project from the KU PKD program project. So I'm happy to take questions.
All right, beautiful talk. I think we have a question already, but I'm going to start my first question. I was wondering if you look at the PKD2 knockout C. elegans models, do they have the mating defect? If they do, if you add the wild type EV back into those mutant lines, can you rescue that mating defect?
Yeah, that's a good question. So what Jack is saying, so the LOVE1 and PKD2 mutants have mating defective phenotypes. And what I didn't talk about was that if we isolate EVs and then expose males to those EVs, they change their behavior. So the question you asked is whether we can rescue mating phenotypes of the polycystin mutants by wild type EVs. We haven't done that, but what we are trying to do right now is to use these mutant EVs to see if they have bioactivity in signaling between animals, and we don't have the answer to that question.
Thank you. So next questions. We have several. We have one question from Dr. Fenchan, and he said, great talk, and how are the EV captured by WAR? Which is great, right? Are the captures the EV requires LOVE1 or PKD2 on EVs, or these EVs are the ecosome or not exosome?
Okay, yeah. Thanks, Vong. So I'll answer the second question because that's answer the question. So the EVs that we're studying are ectosomes because we can see them bud directly from the ciliary membrane. We cannot rule out the possibility from sort of the bulk cultures that we could be also looking at exosomes, but just with the imaging, we know for sure those are ectosomes. And then you asked, how are the EVs captured and by whom? That is like a really important question. We don't know the answer to that. So I talked about Ina's proximity labeling of isolated EVs. She also did the proximity labeling in the whole animal, the idea being that that might be one way that she could get targets of the EVs, and she has some candidates for that. I showed with the transfer of the polycystin EVs to the mating partner that they adhered to her cuticle. So we're testing the hypothesis that LOVE1 or those polycystin-associated membrane lectins could be important for that adhesion. We don't know what they're sticking to on the female's cuticle right now. Those are hard questions to ask. The targeting is the hard part.
Yeah. Next question from Dr. Yu. Do these EV cargo protein associate with the LOVE1's PKD2 or the LOVE1 and PKD2 complex?
Okay, so let's see. So Ina did proximity label. So I showed you the PKD2 proximity labeling. Ina also did the same with LOVE1, pulled out many of the same and some different. We have not done direct, like seeing, one-to-one relationship. Yeah, so I cannot say if they're like directly associated with LOVE1 or PKD2 right now.
Okay. The next question from Dr. Messer. Are both the NTF and the CTF of LOVE1 found in EVs?
Yeah, Robin, that's a great question. So Jonathan Walsh, when he was in my lab, he tagged LOVE1 at the N terminus with M scarlet and the C terminus with M neon green. So this is a genomic locus that was modified. And he was able to look at green and red. So red being like the N term LOVE1 and green being the C term LOVE1, and showed that they actually localized to different parts. So we don't see the N term LOVE1 on EVs that are shed into the environment. We do see it possibly on EVs that are retained in the animal. I didn't talk about that. We need to do expansion microscopy to get a better idea as to whether the N and the C term are associating with each other in the animal. I don't have the answer to that. But there's differences in where they locate, which is really intriguing.
All right, thank you very much, Dr.
Thank you.
Barr. I think that's
Thanks,
end
everybody.
of this talk. All right, I'm going to turn back to the Francisco for introducing the next speaker.
Thank you. So for the last presentation is an abstract. The title is The Nutrient Sensing mTOR Pathway in Renal Cystogenesis. It is Dr. Flavian de Fatima Silva. I hope that I pronounced it correctly. And is from University of Massachusetts Medical School.
So we studied the biology of nutrient sensing. While we may be new to the PKD field, my PI, Dr. Gerting, has been working on mTORC signaling and metabolism for many years and was involved in the discovery of mTORC1 complexes, and we are very excited to contribute to this field and to this wonderful consortium. In fact, we have already obtained many tools from the consortium, and we are very grateful for. And I have joined the lab in 2023 for my postdoctoral training, specifically to work in this project, and we are excited to hopefully contribute to helping people suffering from PKD. So mTORC1 is an important cellular nutrient sensor that controls cell growth and proliferation. And aberrant mTORC1 activation is a hallmark of polycystic kidney disease, but how its activation drives cyst formation remains unresolved. For mTORC1 to be activated, it needs to be recruited to the lysosomes. And this process is organized by the RAG GTPases. The RAG GTPases are essential amino acid sensors organized in a heterodimetic complex. And there are 4 RAG GTPases, isoforms, RAG A, B, C, and D, which forms a complex, RAG AB, with RAG C or D. Briefly, in the presence of amino acids, the RAG complex recruits mTORC1 to the lysosome, where it is activated by RAG GTPase. And when activated, mTORC signals to a variety of substrates, such as S6K and 4-ABP1, that collectively promote cell growth and proliferation. The increase of mTORC1 activation in PKD model has been reported before, as we can see by the increase in phosphat-6 in the cyst lining cells, and the prevailing view is that aberrant mTORC1 activation in renal tubular epithelial cells promotes cyst formation through its established effects on cell growth and proliferation. In this sense, if we think that the RAG GTPases are the central organizers of mTORC1 signaling, if we take all the RAG AB, RAG GTPases in general, we expect the inhibition of mTORC1 pathway and prediction of no cysts. But what we see is when deleting the RAG A and B in the kidney tubular epithelial cells, it promotes a striking cystic phenotype consistent with a PKD model. And by deleting RAGs, we expect the mTORC inhibition, and we see that by lack of phosphat-6-1 in the cyst lining cells, when the cysts start to form by 2 weeks old, and this effect progress by the cyst progression with 4 weeks old and 8 weeks old, where we see low or no phosphat-6 in the cyst lining cells. From this point, we infer that cystogenesis is driven by a mechanism distinct from the canonical mTORC1 pathway. Importantly, the RAG GTPases also play a role in anchoring transcription factors of the MIT-TFE family, such as TFAB, to the lysosomes. When translocated to the nucleus, TFAB promotes autophagy, lysosome biogenesis, and other metabolic process. And one important thing I want to highlight here is when mTORC1 is active, it can phosphorylate TFAB, and which also blocks its nuclear translocation and its effects. Interestingly, aberrant TFAB activation have been linked to cyst formation in other models of renal cystic disease, as BHD and TSC. But this model suggests that TFAB drives cystogenesis by promoting a feedback to mTORC1 hyperactivation, but this mechanism is still not fully understood. We then asked, can TFAB drive cystogenesis in regular AB mutants without mTORC1 activation? And the answer is yes. We see TFAB located to the nucleus from 2 weeks old when the cyst starts, and it becomes more evident with the cyst progression. We also checked by RNA-seq, and we saw overexpression of TFAB target genes, which is consistent with its activations. And to confirm that TFAB is the driver of the cystogenesis, we then generated a triple knockout, deleting reg A, B, and TFAB, and it blocked the cyst formation. Our data support then that reg GTPase is the main negative regulator of TFAB in vivo, not mTORC1. This is supported by the fact when we delete Raptor, a key subunity of mTORC1 complex, we should expect the same results as inhibiting reg A and B. But these mice don't develop cysts, and neither we see any changes on TFAB's lysosomal and nuclear localization as we see in the reg AB deletion. So, and how it is relevant to the PKD study. When we comparing our data with models of PKD1 and PKD2, our data is here represented in red. We see that overlap in gene expression in several pathways, and this overlap is even more consistent with the PKD2 mutations. Also, several genes associated with PKD are upregulated in our model with reg AB deletion. Our data also confirms that loss of PKD2 drives nuclear TFAB localization. And in vivo, we also check that TFAB, in a PKD2 model, we check that TFAB is located to the nucleus while mTORC1 is hyperactive in these cells, confirming like previous finding related to PKD. We have then a PKD paradox. How can TFAB and mTORC1 both be hyperactivated? Understanding this mechanism between mTORC1 and TFAB regulation of cystogenesis is really important, since it can lead to new therapeutic and more specific targets for PKD and other renal cystic disease. In conclusion, we have so far that the reg AB deletion is a new model to study PKD, that TFAB drives the kidney cystogenesis independent of mTORC1, that the reg GTPase is rated an mTORC1 appears to be the dominant negative regulator of TFAB in the renal tubular epithelial cells, and that aberrant TFAB activation is broadly relevant in PKD, and it opened doors to reevaluate mTORC1's role in PKD and the use of mTORC1 inhibitors as a treatment for PKD. So for this project, we teamed up with our UMass Chan colleagues, Dr. Gregor Pazur, who brings his expertise in kidney disease, and Dr. Quang Sheng, expert in reg GTPase biochemistry. And part of this exciting project is being conducted by the graduate student Alex Boucher. And we also recruited a team of exceptional collaborators who will elevate this project with their expertise, as well the useful resources from PKD RRC and TSC Alliance. I would like to thank the organization for the opportunity to present my work in my lab and the funding of TSC Fellowship, and thank you so much.
Great presentation. Thank you so much. I think that we have a minute for question. So 5 minutes. So I have a question for you. So do you see any changes in autophagy or lysosome levels because TFAB is being regulated by your mutants?
Yes. So we have all the gene set for autophagy upregulated in this model. So we are now on the way to explore more these pathways and see how these changes on mTORC and TFAB regulation is related to the development of the disease.
Thank you. There is one question in the chat that says, does PKD1 deletion also lead to nuclear TFEV?
Sorry, PKD1 and 2 deletion. Yes, there are like previous studies reporting the nuclear TFEB in both models of PKD and also in models of TSC and also Burthold-Dubé. So independent of the mutation, if it's on PKDs, TSC, or folliculin, we have the same endpoint of TFEB activation, but we have the controversial backup of mTORC1 that we have in hyperactivate mTORC1 and PKD and TSC, while we have inhibition of mTORC1 in our model. That's why we claim that TFEB is the main driver, but we need to understand what is behind and what's the regulation of mTORC1 in this pathway.
Thank you so much. And I don't see more questions in the chat, but also I think that there are only 20 more seconds, so
Okay.
I think we can end the session here. Thank you so much to all of the speakers, and it was a great session. Thank you.
All right, thank you for all the speakers and to the moderators. This will bring an end to the normal program for the PKD RRC. We have the trainee program. I think you should have received a link to that either in the chat or through email. Also, I think there was or will be soon sent an email around with a survey on Symposium. We would greatly appreciate if you guys would take the time to fill that out. The feedback is really important to us. So again, thank you to all the speakers, the organizers, and IDDK for helping to support this event. Terry, I'm going to turn it back over to you, see if you have any closing remarks.
Just to thank everybody for participating. I really appreciate all the effort that went into the talks, the moderators, the questions, and don't forget to register for the FASAB meeting. And happy World Kidney Day.
Kidney Day,
About
yes.
Kidney Day, kidney disease.
Yes.
Bye, everybody.